Okay, my name is Jennifer Moore-Kucera. I'm an associate professor of soil and environmental microbiology in the Department of Plant and Soil Science. And today I'm going to talk about soil microorganisms as key players in climate change. And so our research, my research team kind of integrates and collects data that then hopefully ultimately can be built into some of the carbon models that are used by climate change scientists. And so we sample soils, and then we try to look at the functional attributes, whether it's carbon sequestration, greenhouse gas production, soil stability, nutrient cycling, kind of you name it, we look at it. And then we try to evaluate the microbial community that's responsible for imparting those functions. And so today I'm going to key in on some of those microorganisms and how they relate to carbon sequestration. One of the things I hope you take away after listening to this presentation is that soil is alive. That stuff you walk on every day, and sometimes painfully call it dirt to soil scientists, is teeming with life. Soils are actually the most diverse ecosystem on Earth, and there are millions of bacteria of hundreds to thousands of different species that are embodied in just a gram, and a gram can fit out on your pinky. Right. So on the left-hand side is a soil aggregate, and this root-like thing that's coming down here is not a root, it's actually a fungal hyphae that helps aggregate and enmesh soil particles together. Soils are combinations of sand, silt, and clay particles, and an aggregate then kind of comes together through the interactions of microbes. Physically enmeshing by fungal hyphae, and then also the activities of bacteria that are decomposing organic matter, release basically sticky stuff that then helps glue that organic matter inside of the aggregates. Why are aggregates important? Well, they resist erosion. The erosive forces of wind, and especially wind here, and also water, can be mitigated by stabilizing the soils through microbial activity. So soils store 2 to 3 times more carbon than that combined by the atmosphere and all of the biomass in plants and animals combined. Unfortunately, about 70% of the original carbon stocks have been lost from cultivated soils. But the good news is that we now know better how to manage our soils, and through restorative practices and alternative agricultural management practices, we can actually put carbon back into the soil. And at the same time increase or at least maintain plant and animal productivity to feed the growing population. And if we do it right, we can actually increase the resiliency of these ecosystems to resist the impacts of increasing intensity and frequency of both flood and drought. But it requires the actions of the microorganisms and understanding how these microbes interact and transform different components of soil. On the right is kind of a terrestrial carbon cycle where CO2 is taken out of the atmosphere by photosynthetic active plants. And then that material eventually makes its way below ground, either through litter inputs, falling leaves, and residues, or after harvest, the material left behind is incorporated into the soil. At the same time, there's below ground biomass through root inputs. And then what happens to that material is that it's acted upon by microfauna, including grazers and predators, and then broken up into small pieces that then the bacteria and fungi act on. What we want to select is management practices that stimulates this microbial activity, but ultimately results in resistant or recalcitrant or stable organic matter that's sometimes called humus. If we can increase the pool of this amount of carbon, then we're sequestering carbon, we're taking it out of the atmosphere and helping to mitigate some of the increasing carbon dioxide concentrations. I'm not going to talk about it, but I just wanted to kind of point out that soil microorganisms are critical for influencing both the uptake and release of 2 other major greenhouse gases, methane and nitrous oxide. Anaerobic bacteria release methane through their actions, and that can just be free-living organisms in the soil or those inside of animal guts such as cows and termites. Luckily though, most soils of aerobic soils actually consume methane, and so they take that methane that's being produced by the anaerobic microorganisms and oxidize it before it's released to the atmosphere. The other big greenhouse gas is nitrous oxide. This is directly linked to the actions of both nitrifiers, so organisms that take ammonium, you can think of that as fertilizers and components of manure. They take that ammonium, convert it to nitrate, and in the process they're releasing nitrous oxide. That can also occur through denitrification, so nitrate then can be reduced, and that means converted into nitrous oxide and released under anaerobic processes. So ag plays a big role, and land management plays a big role in how much of this is being released and how much is being consumed. And so we actually have an opportunity to, not just be the bad guys always producing some of these greenhouse gases, but shifting our management practices to actually reverse that cycle and improve the conditions. Because we still have to eat, right? So we want to do that in the most sustainable way. So today I'm just going to talk about a couple of studies that 2 former PhD students were involved, where we were trying to identify agricultural systems here in the Texas High Plains that increase or maintain plant productivity and get at this increasing resilience and enhancement of ecosystem function. The targeted agronomic systems are called integrated crop livestock. That just means we're bringing animals and crop production together on the landscape. A lot of the previous research by scientists in our department had shown that these systems reduce the amount of water as well as the amount of fertilizers that are input, and while at the same time they're maintaining a profitable plant and animal productivity. And so these ICLs, or integrated crop livestock systems, they incorporate perennial or rotational crops. From an ecosystem perspective, this translates into building organic matter, that's what we want, that stores and sequesters carbon. At the same time, we're protecting and aggregating that soil together to resist wind and water erosion. And it's really driven by the actions of this vast genetic diversity of the microbial community that's stimulating the process and transformation of organic residues into stable carbon and nutrient cycling. So this is on the y-axis, we have fungal operational taxonomic units. It's a measure of diversity. You can kind of think about it as the number of different types of fungi that are there. And then we have 3 different integrated crop livestock systems, and they're compared to the traditional or dominant crop in our area, monoculture cotton. The cotton fields are irrigated. The ICLs differ depending on whether they're irrigated or not. The first 2 components is a perennial native grassland and a rotation of cotton and millet that's not irrigated, so dry land. The next one is a pretty heavily irrigated old willow bluestem Bermuda grass, so a perennial 100% grass, full cover all the time, but irrigated. The next is a crop circle. Half of it is grassland, old willow bluestem, and the other is a rotational corn and whatever the market drives, sunflowers, cotton, and then again the cotton. So what we find is actually by shifting to systems that have a perennial component, we increase the fungal diversity relative to annual row crops, and the lowest diversities were measured in the continuous cotton. So by incorporating perennial grasses or rotational crops, we can actually shift the population and increase the diversity of fungi. So that's great, right? But what does that mean from a functional perspective? Well, we also measured how much carbon is stored in these protected pools. Remember in the beginning I showed you that aggregate, there's all sorts of clumps of stuff inside. If we can choose management systems that enhance or increase the amount of carbon that can be trapped inside of those aggregates, that's a good long-term storage of carbon sequestration. So management systems that increase the amount of carbon that's protected inside these pools is a good thing, and we can help to mitigate some of the increasing CO2 concentrations. So here that is just the measure of how much carbon is inside of those protected pools. And what we find is by shifting from monoculture cotton to integrated crop livestock, even in a dryland, which is the first one, system, we're actually increasing the amount of carbon sequestration potential. That's extremely important as wells are drying up and farmers are transitioning from irrigated crops to dryland. We still need to be productive, but we want to do it in a sustainable way, so it's actually a win-win situation. And then the higher levels are, of course, in those systems that get more water. But even in low water inputs, we're improving carbon sequestration. The same thing I could say, so who's driving, what's responsible for that, is the actions of fungi and arbuscular mycorrhizal fungi. As those guys increase, carbon increases, stability increases. And then the last slide I just want to show you is a study where we're looking at the increase of restoration time in grasslands on diversity. And what we find is that there's an increase up to about 14 years, and then something weird happens, and there's no further increase in diversity. Well, what we're looking at now is going back to look at environmental records at the time of establishment, and how did those conditions, when these grasslands were being restored, influence their trajectory moving forward. So jury's still out onto why that's happening. And then the last thing is just this decrease of diversity from 2012 to 2014. What the difference is 2012 we're coming out of drought. So we're basically shifting from almost air-dried soils out in the field to regular moisture contents. And we actually get a decrease in diversity with this. So we're not sure why. It could be different resiliency of certain taxa bacteria. It could be preferential bacterial grazing by microfauna, but something's happening. And as we go through these cycles of drought and improved conditions and drought and improved conditions, we want to know what that means from the functional perspective of soil. So we're investigating that kind of now as we speak. And then the last thing I hope my program can contribute to is we're trying to seek funding to evaluate climate projections are indicating that drought is going to increase in intensity and frequency, and with drought comes dust events, as we have experienced recently here in this region. So we want to know what's carried in that dust, what microbial communities, we know bacteria, viruses, fungi, all sorts of things can be carried in the dust particles, as well as potential contaminants, including heavy metals. How does ag influence that in management, and what's happening at this urban ag interface? So with luck, we'll get some funding to study that. And just want to thank a whole suite of collaborators here at Texas Tech and elsewhere, and of course all my undergraduate and graduate students over the last 6 years. So thank you.
Questions?
Yes,
Carrie.
On your restored grasslands,
yes.
Was that restored short grass prairie, or was that CRP, or?
Yeah, so there's CRP, and part of the other story is that 14 years and younger, that's when the NRCS agents became more aware of diversifying their seeding mixtures into less monoculture, non-native species to a more diverse group of grasses. So that's certainly a contributing factor, but we don't have old sites under the mixture of grasses. So we actually hope to be able to track these systems in the future to identify those 14-year fields getting, increasing with increasing diversity due to above ground diversity.
Are you going to have any problems with that, with losing CRP?
Yeah, that's one of the main problems is Texas leads the nation in CRP, restored grasslands, but they also lead the nation how much land is coming out of CRP. And so we actually have another study where we have fields that were recently converted back to cropland production. We're tracking those changes over the short term, so we'll have more information in a couple of years. Yes?
Just wanted to clarify something. So
yes.
You said drought leads to less diversity in the soil. Is that true?
Actually, the drought, the samples collected under the drought conditions had higher diversity than the samples collected under the sort of improved conditions, if you will. Mm-hmm. Whether that's a short-term phenomenon or whether that's just sort of like this sort of initial disturbance that kind of keeps the competition at bay in the drought conditions, we're really unsure of, but the data kind of parallel another study back from 2011 during the drought. Same kind of thing happened with the bacterial populations. Diversity is higher under the worst conditions, but interestingly, fungi were actually higher under the improved conditions. So another component of that study, we have a whole sequencing data set we're analyzing now. What's happening to the fungi? We think they're a little bit better predictors and a little bit more able to withstand drought and impart that resiliency on ecosystems. So there's a whole nother layer of the story that I didn't share with you, but from a bacterial perspective, diversity actually is higher under drought. Fungi seems to be higher under the better conditions.
And is there, I'm sorry,
Okay.
any relationship between how much carbon is trapped during drought?
Uh-huh.
I mean, is there a change, or can you?
Carbon takes a long time, organic matter takes a long time to change, kind of decadal time sequences, so we often use more sensitive indicators, different pools of carbon, and we're looking into that to see how that is reflected in this restoration program as well.
That was what I was wondering too,
Yeah.
is there a really good correlation between diversity and carbon storage, or is it kind of fuzzy depending on the other factors?
Well, in our other, in the study from the beginning where I showed you, let's see, this guy, right? So the systems, the perennial systems from a fungal perspective, the higher the diversity, were the same sites that had that protected carbon pool, the carbon sequestration potential. So there seems to be a direct relationship between fungal diversity and carbon sequestration. This jury's still out what's happening with bacteria and whether, how they interact with the carbon story. But they're certainly important as well. But fungi seem to be, and the thing is that bacteria are way easier to evaluate molecularly and otherwise, and so now we're really excited to be able to contribute that body of knowledge from the fungal perspective. The tools are becoming a little bit easier to work with and interpret, so that hopefully will help try to answer some of those questions.
Great, thank you very much. Oh, one quick question from Kat.
Yeah.
Do these
I'm curious
systems
on the
control
terminology.
for
Cotton.
the
Wait, hold on.
different types
Okay,
of
two
soil?
questions. Yeah. Wait, what?
Do these different systems control for the same type of soil?
Yes, they're all the
going on?
same soil series. These are clay loam soil, a palmate clay loam. So they're finer textured than some of our other soils, but they're all the same. They're actually in the same region. Most of them are on the same site.
So follow up if I might. So if they're all controlled, and you know that there's the fungus is preferred during the wetter times, then if there's different soils in the region, can this be expanded to determine if there's a site suitability preference for fungi versus bacteria in different types of soils in the region?
Probably. It would take a pretty intensive sampling effort, but yeah, in general, fungal have higher carbon efficiencies, and so systems that support a higher, fungal bacterial biomass ratio tends to support more carbon sequestration, and that's kind of been documented in the literature. Did you have one more?
Oh, a quick question.
Yeah.
Consider cotton and corn, they're monocultures.
No, yeah, well, they can be, right? But these guys are monocultures. This was a rotation. So corn was the year that we sampled, but it had been under sunflower, it had been under cotton, it had been under a couple other things. This guy was a rotation of millet and cotton.
But what monoculture just means it over and over again the same.
Monoculture, same thing over and over again.
So the natural grasses, that's over and over the same use, but is that considered a monoculture too?
Well, it's a perennial system, so I guess it's a monoculture if it's just this one. But perennial seems to be a bigger driver than annual when it comes to diversity of what's there. So this guy is actually perennial native grassland. This is a mixed species grassland composition. So it didn't seem to matter from a fungal perspective whether it was a monoculture grass or whether it was a mixed grass. It was just that perennial cover that seemed to be driving it. The more stable, right, like tillage and that kind of operations, that chops up the fungal hyphae and it disturbs them more than bacteria. So anything you can do to keep it calm, basically, is good. Yeah.
I asked John a question and I don't.
Did he answer? Because that would be awesome.
No, it is what happens when you use herbicides or pesticides or fungicides on the land. How long does it take, if you will, the fungi to recover microbes in each of these areas?
Right, yeah, that's a
depending on the plant?
great question. It depends is the cop-out answer, but so a lot of organic production is looking at some of those relationships where they don't use the same kind of, chemicals to ward off diseases and herbicides. But, there's a lot of, I haven't personally looked at it, but that's a great question and would be worthy of some future studies.
Would you guess it would be
So-
like months or years or decades, or what kind of?
It depends on the chemical and its persistence. Some studies have shown, for example, glyphosate pretty short-lived, and then other studies, depending where you look, can show long persistence that does alter the community over time. And
You have to say, that persistent aspect has to do with the amount of organic matter in the soil.
That's true.
And the stability of the pesticide and herbicide. So it's a dual edge. The more you
Yeah.
add it to a soil that's sequestering carbon, the worse off it is.
And as the crops become, or weeds become more resistant to these chemicals, and then we
More
transfer to other chemicals that are longer persistent or more volatile that can move off site, and that raises some more questions. So it's job security.
What is the difference between those two carbon data?
Nothing. Two completely different geographically isolated fields. Same system,
Same
just
system.
different locations.
That's good. To keep fair.
Mhm.
All right. Unfortunately, we're going to end. Who knew that this would be the most fascinating discussion?
Microbes rule the world.
Exactly. And soil is
Alive.
alive. Yes.
It's not dirt.
And it's not
Right.
Thank you.
All right. Let's thank all three speakers. And
That
please
was a famous